Thank you, Adrian, for inviting me to speak at this really exciting group. And let me just now share my slides. Okay. So I have been invited to speak to you today about biosampling, deep phenotyping, and data sharing in relation to the NeuroDev study, which I am the research director of. And I thought the most compelling way to do that would be to tell a story of a case that generated quite a lot of excitement because it was the first case that enabled us to contribute to a new genetic discovery. So I work alongside 4 principal investigators, Elise Robinson at the Broad in Harvard, Kirsty Donald at the University of Cape Town, Amina Abubakar and Charles Newton at the Kemry Wellcome Trust, and both have secondary affiliations as well. I started this work on NeuroDev in 2016 from the very first days of the study. And I, at that time, was employed in Boston from the Broad. And 2 years ago, I sidestepped over to Oxford, which is where I am now employed. And Oxford has an administrative role as a partner to Kemry on the study. So I understand that Kirsty spoke to your group last year in Belgium on largely on the phenotyping of NeuroDev and on the ethics surrounding that. And so I'm going to do my best not to repeat what she's already told you. Instead, I will focus in on biosampling, deep phenotyping, and data sharing, and use the case of a child that we're calling Luke as an example of the study design in action, with the hope of bringing that to life for you. And then I'll finish up by describing where we're heading in the next few months. So starting with a bit of context and our case, these are the 2 hospitals that we recruit from in Cape Town. They are Tygerberg Hospital and Red Cross Children's Hospital, which together have a catchment of the entire Western Cape province, which covers about 2.2 million children. Red Cross is the largest children's hospital in sub-Saharan Africa, and Tygerberg is the 2nd largest hospital in South Africa. So together they are both powerhouses of practice and research in the continent. The population that we are sampling from is largely Xhosa and mixed ancestry. And so we conduct our assessments in 3 different languages: Xhosa, Afrikaans, and English. The overall design is a case control study with a nested family study. So when we speak of participants, we're talking about children with neurodevelopmental disorders as defined by the DSM, and age and ancestry matched child controls, and where possible, the parents of the cases. So as of the lockdown in March, these were the numbers we have reached in South Africa, with close to 1,000 participants, of whom over 300 cases and nearly 200 trios, which is almost double the 4-year target that we had going into the study. So that was really excellent news from the start on trio ascertainment. So Luke is participant, in fact, 859-442-065-3601, totally de-identified child. But I'm going to try to bring him back to life for you a little bit. When he joined us, it was June of last year. He was almost 2.5 years old, and he didn't yet speak. He had actually just learned how to walk a few months prior. He was a late walker at 21 months, and he was a short child with a large head. He had been previously diagnosed clinically as having global developmental delay, which is how he got into our study. And his parents complained that he frequently would wake in the night and that he would get very distraught and rock a lot. His parents came to us from a relatively middle-class background. They lived in a house with running water and electricity. Luke's mother was a homemaker, and she cared for a household of 8 people, and his father worked full-time. He had one other sibling who was developing normally. And so the parents didn't really understand why it was that Luke was struggling so much and came to us quite desperate for answers. So what did we know going into the study about what might have caused Luke's condition? In 2017, when we started this process, the Deciphering Developmental Disabilities study, the DDD study, had just published a landmark paper pointing to 94 genes involved in developmental disorders, where damaging de novo mutations would typically cause developmental delay. Now, we didn't have any screeners for those genes at the time that Luke was enrolled, but even if he had been screened, we now know that he didn't have any of the known genetic causes of developmental delay. 1 year later, we received this very exciting email from the Matchmaker Exchange hub, which told us that they believed that Luke's condition was matched to that of 7 other individuals, and that the most likely genetic culprit was the AGO1 gene. So the timeline from Luke's enrollment to the discovery of the AGO1 gene was almost exactly 1 year, which is startling, but I promise you I didn't make these dates up. Here you can see the sort of steps along the way from his sample being shipped and shared with the NIMH to passing QC and sequencing, and then uploaded to the Broad Center for Mendelian Genomics, and finally analyzed and shared. And I'm going to walk you through that process in more detail. So what are the 3 major components to making a discovery like this? I want to highlight first the biosampling for maximum use, next deep phenotyping, and finally swift data sharing. So starting with biosampling, what do I mean by biosampling for maximum use? I want to focus on 3 main elements of biosampling. One is broad consent, the second is biobanking, and the third is generating cell lines and, where possible, iPSC. So to start with just the nuts and bolts of what samples we are collecting. We're collecting 10 milliliters of blood from all of our participants. So Luke gave 10 milliliters of blood and a pilot saliva sample because we're also trying to see whether we can get good quality DNA from that. But the blood sample enables us to generate cell lines, which is a key part of our design. So what happens after collection? Luke's sample would have gone through 5 different laboratories, 2 of them in South Africa, 3 of them in the United States, and those are research laboratories alone. First, it would have gone to the NSB, which is the NHLS and Stellenbosch University Biobank. And there it was processed and one of the tubes was spun to generate cryopreserved lymphocytes. Then it was shipped with one tube of whole blood and several tubes of CPL over to Rutgers University, where DNA was extracted, and it was stored at the NIH repository and genomics resource, and then later extracted from there, and an aliquot was sent to the Broad, where it was sequenced. So slowing down, starting that process from the top, the first step to biosampling is consenting the participant. So this is an excerpt of the consent form that Luke's parents would have seen, and it highlights the concept of broad consent. And it's, despite the name of the Broad Institute, we do not have a monopoly on the concept of broad consent, but it is something that the Institute feels strongly about. And at the time when I started at the Stanley Center at the Broad, they had just published their commitment to genomic data sharing, which I've shared a link with on the bottom of the slide. And it can be a hard concept to get your mind around broad data sharing, but we were lucky in the context of our South African site to have the precedent of the H3 Africa genetic studies, which had familiarized people with this concept and with the utility of it. And so we were able to get through this language that is really quite bold, that says not only that the samples will be available to scientists all around the world to study any research question, but also that we don't know what the future uses might be. So the next way in which we have collected samples and made Luke's sample maximally useful is by depositing it for biobanking at the NIH repository. The NIH repository is governed by a data access committee, but the samples are made freely available to really anyone who has scientific qualifications and a rational research question. So we're hoping that by doing this, the NeuroDev samples will be able to be analyzed by a much broader community than the NeuroDev investigators and their affiliates, and including potentially people from the MINDDS network. So next, the third way that I want to highlight in which we've maximized the use of our biosamples is through cell line generation. And it's quite a cutting edge thing to be able to generate cell lines from participants with neurodevelopmental disorders in an African context. And that was really possible thanks to two main ingredients. One is that we had precedent of this through the schizophrenia in the South African COSA population study, or the SAC study, of which Dan Stein was a principal investigator in South Africa. And through that SAC study, a partnership had been established between the NSB and Rutgers laboratories. And the very smiley face you see on the left there is that of Carmen Swanakwel, who manages the science and operations of the NSB and is co-investigator on that biobank. And to your right, you have Michael Sheldon, who is an associate professor of genetics at Rutgers and head of the NIMH studies that are being managed by Rutgers. And that partnership was really essential because we knew that this work could be done. It could be done to very, very high standards, and it could be done swiftly. And so, you know, I think we were very lucky to be able to step into the shoes of the SAC study and benefit from the expertise of NSB. Because there are a number of people in the MINDDS group who have a particular interest in iPSC, I wanted to highlight some of the questions that were raised by participants at the time of being consented for stem cell research. And just to clarify, we are generating at the moment cell lines from all of our participants, but stem cells would be created on a selected basis, subject to additional funding. So the questions raised by participants fall into roughly three categories. There were questions about the mechanics of stem cell generation, and particularly why it's happening in the US and not in South Africa. There were questions about the benefits and who they would go to and whether they would be monetized. And also whether they would be useful in treating conditions that are frequently seen in the South African context, like HIV and cancer. And then there were questions around the risks, including questions about the cloning of children. So really the questions ran the gamut, and I think that they illustrate in some ways just how novel of a concept this is. We can come back to those if you have further questions at the end. The last step in the biosampling line is the sequencing. And Luke's sample was exome sequenced and genotyped at the Broad using the global screening array. By doing that, we made his and his parents' sample comparable to all the other large-scale genetic studies being run out of the Stanley global collections, which are all also using the global screening array. So traditionally, genetic studies have had a very light touch approach to phenotyping in the interest of creating the maximum sample size. But the NeuroDev study took a slightly different approach and decided to deeply phenotype its participants in the hope of being able to contribute to fine-grained descriptions about phenotypic variants in people with newly diagnosed rare genetic disorders, and particularly among African individuals with those disorders. So the first step of phenotyping is sample ascertainment, and this is just a quick overview of the inclusion-exclusion criteria. Luke was able to join because he had a pre-existing diagnosis of global developmental delay, and he did not have a primary motor disability. Luke's, as I mentioned, was nonverbal at the time of the assessment, so the information we got about him was principally from a combination of parental self-report, direct observation by clinician researchers on our team, and then a consultation with the medical records available to us. Data were directly entered into REDCap and uploaded on the same day. We phenotyped Luke along 7 different domains, which you can see here, starting with cognition, also looking at traits of autism, at behavioral disorders, at facial dysmorphias, and also the hands and feet, his demographic background, his socioeconomic background, and his neuromedical history. These are the specific tools that we used, and I know that Kirsty went into this previously with your group, and so I'm not going to dwell for too long, but I do want to note that parents were cognitively phenotyped on this study, which is a real strength of the study because we note that if a parent had a particularly high cognition, it could be that the child had a decrease in their cognitive ability that wouldn't necessarily appear so large, but could in fact be large, or inversely, if the parent had low cognition, if the child's decrease in cognition was 1 standard deviation, it could appear significantly worse than otherwise meets the eye. The cases on our study were being phenotyped with the 3DI brief and the social communications disorders checklist, the SNAP ADHD, and the CDCL. But otherwise, cases and controls were all photographed. They all received neuromedical questionnaires, and they all took a cognitive test. I've just realized that I omitted the cognitive tool that's used in the small children, which is called the Multino. So what do you do once you have that data? How did we get from Luke's data in REDCap to the AG01 discovery? The first step was sharing it with the Broad Center for Mendelian Genomics. I'm going to focus on the example of Luke and look at 3 of the places that we've shared data with to get to an answer. So what did we share with the Broad CMG? We shared 99 exome trios, of which Luke was one of them. And we shared the information that we collected in REDCap, coded in HPO terms. So that's the human phenotype ontology terminology. We shared that data in January, and we used a platform called Seeker to do so. And the result was that that team analyzed the data, identified some likely pathogenic variants. They also identified those who had diagnosable conditions that are already known. And in the likely pathogenics, we continued the data sharing. How did we get to likely pathogenic? These are the filtration parameters that were used, and this is not my expertise, so I will leave you to read these since you are likely more familiar with this process than I. My background is in health services research. So from there, we shared our hypothetical candidate gene, which was the AG01 gene, on Matchmaker, and we shared it together with a number of phenotypic traits. And on the same day that we put that gene on the data sharing portal, we received a match to a researcher in Spain who was leading a case series paper already on this particular gene. And so we were able to add the NeuroDev case to 7 pre-existing cases of AG01 de novo mutations, and we're now in the process of contributing to a paper. In addition to the HPO terms, which are categorical terms, even though we collect our data dimensionally, there was an interest in sharing the photos that we had taken for NeuroDev. So Luke's photo was the first photo to be shared out of NeuroDev, and it was shared using Face2 Gene, which is an application that allows you to securely share identifiable information. And Face2 Gene also allows you to use computer algorithms to analyze these conditions, and we plan on uploading all of our diagnosed cases to the Face2 Gene portal so that they will become better at recognizing African individuals or individuals of African ancestry with these conditions. So where do we go from here? The next big piece of work that we're quite excited about is returning the genetic result to Luke's family. We have not yet done that, and we are finalizing the consent form that we will do that. We already have IRB permission to do a return of results, but we're fine-tuning the details of it because we want to be sure to get it absolutely right. But I think that in terms of maximizing the impact of this finding, there's no greater impact than feeding it back to the family. The next step will also be to sequence our first full batch of NeuroDev participants, and that will be across South Africa and Kenya. We'll be sequencing our first 500 cases in the next several months. And so that will enable us to continue adding new candidate genes to Matchmaker Exchange and hopefully discovering many, many more variants. And of course, we will continue working on publishing these results. The AGO1 paper is underway, and we have 7 case series papers also in progress at the moment, and are working towards the first cohort paper on our first 99 trios, which combine data from South Africa and from Kenya. We're also looking forward to relaunching our study enrollment. In South Africa, we have redesigned a number of key pieces of our study so that we can remotely capture data, including using e-consent and telephone interviews and remote saliva collection. And what you see on the right is a video to help illustrate the consent process, which is otherwise a 10-page document and can be hard to deliver over phone. We'll also be continuing our data sharing, so Luke's genetic data will be shared on Anvil and dbGaP, the 2 NIH portals, as well as the NIH Atlas of Human Malformation Syndromes in Diverse Populations, which is an open access online website that provides images of children with developmental delay in populations that are not otherwise seen. And we will also be sharing data on controls from the NeuroDev study and contributing those to the NOMAD database, which currently contains over 125,000 exomes, I believe, from normal population. So that is NeuroDev in a nugget. I want to thank the funders of the study, particularly Ted and Rhoda Stanley and the Stanley Center for Psychiatric Research that they created within the Bird Institute, as well as the NIMH and the NIMH repository and the Simons Foundation for Autism Research. I also want to thank these individuals. This is a picture from several years ago when the study was just starting up in Cape Town. And a number of these people are on the call right now, so thank you.
Wow, and thank you. Thank you. It's really, really interesting. Okay, well, we'll open everything up for a discussion, but so people put your hand up virtually or write a question down. But I guess the first thought I had is how far will you go? Is this going to go for a long time? Would you go back and resample people? Is that part of the plan as well, or is, what's the ultimate scope in the study?
So it's we certainly have consent to resample and consent for recontact. Resampling in the South African context is, we're trying to get around that in part by generating cell lines. What sort of resampling are you thinking about?
Well, I was actually just thinking if you wanted to go back and say, look longitudinally really, and ask the question of how have things changed. Okay, there's a lot of other things going on, but you know, have the conditions changed? Have they, you know, have they morphed into different things, you know, or even just new technologies? I guess that maybe like brain scanning things that you might have to do locally rather than in a centre.
Yes, and I know that your the MINDDS group has a sort of a working group on cohort development, and so I can see how that could be of interest. I think it's absolutely, we've tried to consent people so that, you know, every possibility exists, subject to their reconsent. And I think what we're finding at the moment is that we have very eager parents. They're very motivated, and you know, that's demonstrated by this trio uptake that's much higher than we anticipated.
Yeah.
And so I wouldn't be surprised if the case families would be amenable to participating in further research. I don't know, the blood draw itself is not the favorite part of the study, as you might imagine, and I think that both the families and our assessors are somewhat heroic in being able to draw blood as they do from these children.
Yeah. Okay. So Yassir has a question. He's just written down. If you might as well read it. You might ask a question yourself rather than you read that. Mute.
Hi, Victoria. Thanks for the talk. Very nicely illustrated about the different phenotypic criteria that you're implying. So as you're recruiting the patient, what is the average age of the people that you're recruiting? That's my first question. And the second question I have is, as we know that the African genome is quite unique in a sense that they have an average 10% more genome than the rest of the population of the world. So I was wondering, is there any unique behavior that has been identified during the phenotyping?
Good questions, Yassir, thank you. The average age in South Africa is quite young, and that's because it's about 3.5. And that's because we're dealing with a really excellent tertiary care center where people go early. In Kenya, it's a much higher age average, closer to 8, and I can give you the exact numbers afterwards. But in terms of the specific behavioral traits, I'd say, you know, we're not, our study design is not a population study, and we can't really speak to prevalence. But what we're seeing in our clinical sample, in our clinical ascertainment, is that we have a lot higher levels of intellectual disability associated with autism in this population, which makes it more likely to be associated with genetic syndromes. But that's about all we can really say at this stage. And even since these are rare genetic disorders with only about 8 cases, it's not even really possible to say how they vary phenotypically by ancestry because it's such a small sample. But you know, we hope at a later date to be pooling our data with other consortia, including the Autism Sequencing Consortium, which Elise Robinson chairs, also the autism group within the PGC, and to do some common variant analysis. And that might be able to reveal some sort of finer tuned answers to your question.
Thank you.
Paula, you've got your hand up. You revealed yourself. Yeah. Here you are.
Hi again. This is Paula with me, and she raised the hand, but actually I had a question which I had sent to Ilka, but it was for everyone, particularly Victoria, because you said that on relaunching the study enrollment, you did remote capture and used an e-consent. What sort of consent is this, and how does it differ from the original informed consent?
So it's the same document, but REDCap, and I'm very glad to see that Alice Galvin is on this call to keep me honest. Alice, feel free to chime in if I'm misrepresenting this. REDCap enables you to do a signature that is authenticated. So rather than requiring, say, a printer or a human person or a mail system to courier you your signature, this way you have an instant verifiable signature that is sent online.
So it's like a digital, it's a digital signature, basically.
It's a digital signature, yeah.
You send a document by email or something, and then you put down your digital signature.
Yes. Alice, do you want to, is it by email? I don't believe it's by email.
It is a link, a web link that would be sent by email. Yeah. So that way the participant is able to view the answers that have been sort of put in by the researcher, so they can confirm, and then the only fields they would have to fill in are the signature and the date, and that gets captured directly into REDCap. So there's no sort of external files that are sent between email. It's just a web link that they receive.
So it's basically a secure connection to enable quite complex data or information to be.
Yeah.
So that it cannot be easily shared or seen by anyone. Okay.
Exactly. It gets around data security concerns, which are rife with this type of research.
Okay.
So that's universally applicable in the sense.
Yes.
Same town. It doesn't have to be random.
We'd be happy to send you more information about that if it's of particular interest.
Yes, I would be happy if you do that. If you send the information to Adrian, he can send it to me, or you can send to my Gmail. I will look for your name here and try to write the email here. Okay. Hopefully it will get to you.
Okay.
Thanks.
Great.
Right. And to go back, just to sort of pick something up from Yassir's question, and I know you won't be able to answer this question yet, but it's worth thinking about it. So you must pick up sort of common CMVs. I mean, they're all rare, but you must be picking up rare variants that you'd see elsewhere in the world. And do they manifest themselves in the same way? I guess you haven't got enough data yet. I'd be interested to know if you've got any insights into that.
I mean, that's that's the jackpot question, and it's very hard to say at this stage. I mean, I think we hope maybe in several years to be able to respond to that question.
Yeah.
But, you know, to contextualize it, Charles Newton likes to speak about how he learned about genetic syndromes using a German textbook that had lots of pictures of Caucasian kids with dysmorphias. And he even pulled up the textbook on one occasion. And the NIH Atlas of Human Malformations in Diverse Populations, which is a mouthful, is sort of one answer to that, which is to try to create that textbook with a broader population group. But Face2 Gene is still another step further in that it's getting beyond the kind of trained human eyeball and trying to put computer algorithms to sort of map features, not even individually, but sort of as a whole. And so you move away from, you know, specifics about the eyes being so far apart, and instead you get this kind of heat map of the face. And then it generates, you know, the hope is that once it has enough information, that it can start generating model faces of people with given disorders, but it needs to have the right input. So at this point, it's not clinically useful for our populations, but we hope to contribute some data to help send it in that direction.
Yeah, and there are projects in Europe and elsewhere that are similar. But have you collected enough genetic data just to know, for instance, whether the frequencies of, you know, something like 22q11 is the same or different or distorted, or is it still quite down the line?
I think so. We've started our CNV analysis of the first 99 trios, and we're finding the same common CNV pathologies. Again, prevalence is just really hard to say because we're not an epidemiological cohort. So unless you're assuming, you'd have to make a big assumption that somehow you're genuinely catching the entire 2.2 million. But the reality is that you probably, you know, who ends up in the clinic is very much influenced by the socioeconomics, the distance from the clinic, and so many other factors. So it's just, we just can't answer. It's a really interesting question, but we can't answer it, unfortunately, yet.
I was just wondering whether, I mean, exactly, maybe the context is too different, but whether it's at least is indicative of, you know, sort of biological differences rather than, or biologically based differences rather than sort of socioeconomic or just actually how the health service system works, which of course is linked to that last thing.
I mean, one big sort of environmental risk factor that there's less of in South Africa is parental age, because parents are tending to have kids at a younger age. So you might expect to see somewhat lower rates of de novo mutation because of that, but that's really all I can meaningfully say.
It's curious, that's all, because there's a sort of, as a higher level of a piece of information there, isn't there? But it depends on scope, I suppose, the amount you can collect quickly.
It's a good question. You know, it's the question we all have.
Well, of course, absolutely. Yeah, it is the million-dollar question in many ways, isn't it? And that's what you see, and you don't know what's generated the differences. Yeah. So, and the data sharing. I mean, we're obviously interested in data sharing and how we do things and material sharing. So Matchmaker. So that is set up as a sharing platform, or is it actually a sort of genetic platform with other things added to it. Again, you may not know the answer to this, but I'm just curious.
It's a sharing platform. I'll just take you back to one slide in my deck.
I think we're pretty interested in that. I mean,
there were,
right?
yeah, I wasn't sure how much I was preaching to the choir, so I didn't go into.
We're probably aware of what the issues are.
Tremendous discussion. But this, let's see if we can. So is it going to like that? Let's try again. There it is.
Thanks.
So this shows you the different nodes of the Matchmaker Exchange. So the Broad has one called Matchbox. The Sanger has one called Decipher. Hopkins has one called Gene Matcher, and so forth. And each of these has a, I think I don't know whether they all use Seeker. That's something I can't answer for sure. And I know that Seeker is now being overhauled, and there's a new interface that's being created. But basically it's quite straightforward. It's literally, from the phenotype perspective, more or less an Excel table of HPO terms. Yeah. And then because we had sequenced our data at the Broad, they just got the entire exome sequence handed over to them. And then there are all kinds of amazing things you can do from within it as an analyst, which are quite exciting to see. And if you are interested in that aspect of things, I'm happy to put you in touch with Emily O'Hare, who's been one of the people analyzing the data for us there, or really with any of the people. Anne O'Donnell Norrie has been, she is heads up the Broad Center for Mendelian Genomics alongside Heidi Wren. Yeah,
no, I think we're very interested in it, although in the sense of, and the example would be, suppose you wanted to incorporate the data you get from face-to-face into that, which sounds like, oh, just do it. But of course, how you do that, how you encode two completely different sets of data gets quite interesting then, doesn't it?
What do you mean by the data from face-to-face?
So you connect well, okay, this is maybe getting to the issue of what it is. So you've got strong connectivity through the genome. It's pretty much the same sort of data. It's about matching, as in the name. But then if you want to start hanging phenomic data on it, well, you've got HPO and other aspects of that. But as you said, it's a bit like a list. But if you wanted to put other modalities, for instance, morphometrics and the face morphometrics, that data will be different again. And so how you interface with two of those gets sort of more and more complex. I'm just sort of curious. I mean, that's one of the problems, I guess, that we're grappling with the group, is how do you take all these sort of different types of data? Standardizing it is one thing, which is obviously incredibly important, but then how you figure out how to share that and connect two different types of data together in a way that's sort of useful is probably the biggest question of all, really, isn't it? So,
yeah, well, I mean, I can say that Matchmaker Exchange has worked beautifully for CNV variant analysis. So the only thing that the platform doesn't allow is photo sharing, and that has been complemented quite nicely. Well, there are two things, sorry, I'm conflating two things. The Broad Center for Mendelian Genomics is platform, and the Matchmaker Exchange platform. But the Matchmaker Exchange platform is chiefly about matching candidate genes that have become candidate genes through a careful scrutiny of of variants, and looking at OMIM and looking at ClinVar and looking at all sorts of other gene registries. You're right that it's principally about creating that connection. and then the sharing of the data sort of happens, in our case, in any event, in this case of the AG01, once the connection is made, then you're just firing emails. Essentially. And there may be a better way. There may be a better way.
You sort of have a sense there's a better way, but I don't know the answer either. That's why I'm curious. You sort of sense there might be a way of doing something really neat with it. But not yet. And where do you start? That was sort of what was in the back of my head. Because I think there is. I mean, you never need one platform. You can have as many platforms as you want, but there is a sort of global sense of how do we make that work beyond DNA. So DNA is a nice portable.
I'm conscious we're at the end of our time, and that some of the colleagues in Africa, it's quite late in the day. So I want to thank you so much, Adrian, for giving us the opportunity to share the NeuroDev experience with your group. And I really hope that it will just be a continuing dialogue, because there's only so much you can say in an hour. And I'm glad that this isn't the first, that Kirsty came before, and I hope that there will be others to follow, because there really is a tremendous amount of overlap, particularly as we're starting our CNV analysis now.
Yes, particularly that actually. But no, it's been fantastic. It's actually been really, really interesting to hear about it. And I was sort of vaguely aware, but now I'm a lot less vaguely aware. And I think you did a really brilliant job in just sort of putting it together. It was really impressive, but really good. So yes, I really hope we can sort of, keep together, if you like. I mean, you guys are always welcome to get involved. I mean, one of the silver linings of this difficult situation now is that we're not traveling. Therefore, a lot of the limitations have gone. As long as you can get a connection, it's a great opportunity to really reach out and share our information, knowledge, and thoughts. I think. But yes, I think on behalf of all the Minds people and anybody else, actually, because I know there's other people we've dragged in. Been really brilliant to hear from you, actually. Thank you so much, and thanks to the NeuroDev team who were on the call. I really appreciate your being there. Excellent. All right, so thank you. Bye. And hopefully see people again towards the end of the month, which is probably, time flies when you're sitting at home, doesn't it? Okay, bye everyone. Bye.